Hello students, I am Dr. Leena Mishram, working as Assistant Professor in Zoology in Mahatma Phule College, Panvel. Today we'll learn a very basic but very important concept of molecular biology, and that is central dogma of molecular biology, which is also called as central dogma of life. Now we all know that the basic unit of life is cell. So this is the structure of a cell: nucleus, and the nucleus has chromosomes. These are the chromosomes. Now in case of human being, we have 46 chromosomes, that is 23 pairs. We also know that these chromosomes are responsible for all the different traits or all the different hereditary characters in an individual. That is, if I look tall, that is because of these chromosomes. If my eyes are brown, that is again because of these chromosomes. If I am fair, it is because of the chromosomes. So all the expressions or all the morphological characters, all the physiological characters are because of this chromosomes. All the expression is because of the chromosomes. Now these chromosomes are made up of chromatin network that is highly coiled around histone proteins. So when we study the structure of chromatin, we know that it is having highly coiled DNA. This is the structure of DNA. And we also know that a short segment of DNA is called as a gene. The short segment of DNA is called as gene. A stretch of DNA is called as a gene. This gene gives an expression. This gene gives a trait. This gene gives a character. This gene gives a specific morphological expression, or whatever your eyes can see of a person, a character, everything is given by this gene. Gene is a segment of DNA. Now this gene is responsible to synthesize a protein. What I said is, if my eyes are brown in color, it is because of a pigment which is a protein which is giving it brown color. If my eyes are gray color, it is because of again of a protein. So how this gene expresses itself in a pigment or in a protein, and that is given in central dogma of molecular biology. So when we say this gene responsible for protein synthesis, how it is possible? So gene responsible for a character, for a trait, for the synthesis of a protein which expresses itself. The gene expresses itself in the form of protein. Expression in the form of protein. So this flow of hereditary character, or this flow of information from a gene to a protein, is called as central dogma of life, or it is called as central dogma of protein synthesis. Okay. So when you talk about DNA is double-stranded structure. DNA is a double-stranded structure. It is a genetic material of all the living organisms. It is a genetic material. So all the genetic information is stored in this DNA. It is stored in the DNA. Few viruses also have RNA as a genetic material. For example, retrovirus, HIV virus have RNA as a genetic material. Now this DNA is made up of a double-stranded structure. We know that there are nucleotides having base pairs, So whatever information it has to transform, it is stored in this coded language, and these are the nucleotides which are arranged in a different manner to store all the genetic information. This DNA is transcribed into RNA. How? How is it possible? So this DNA is transcribed into a single-stranded structure where the thymine base pair is replaced by uracil, and it is synthesized or using DNA as a template. This RNA is synthesized. Specifically, we will say it is messenger RNA. Okay, so DNA is present in the nucleus. The DNA from the nucleus will be forming RNA, and it will come into the cytoplasm. See, protein synthesis will take place only in the cytoplasm. It will not take place in the nucleus. So all the synthesizing machinery, the protein synthesizing machinery, we call it as ribosomes. They are present in the cytoplasm, and hence the synthesis of protein will be carried out in the cytoplasm. So the DNA which is present in the nucleus, whatever signals, whatever information it has to get, it will get, and then it will get activated. The gene will get activated, and it will pass the message in the form of messenger RNA from the nucleus to the cytoplasm. So from the DNA, it has transcribed into messenger RNA. We say it is called as transcription. So all the coded language, all the genetic information which is stored in the DNA, it will be transcribed in the form of messenger RNA for a synthesis of a particular protein. Okay, so this coded language is decoded in mRNA. So mRNA will have codes now. Okay, they are called as triplet codons. It will always be in 3. So this is one code, this is another code. Okay, so all the coded information from the DNA will be carried to RNA. This is messenger RNA. Then it will be again decoded to form a protein. It is now called as translation. Now we all know that proteins, they are biomolecules. What are they made up of? These biomolecules, which are also called as macromolecules, what are they made up of? They are made up of amino acids. We know that there are 20 standard amino acids which make up all the proteins which are present in our body. Proteins play very important function. For example, if you say a very important function of a protein, proteins are enzymes. Proteins are hormones. Proteins are present on the plasma membrane. That is, the plasma membrane have proteins which have lot of function. There are carrier proteins, there are transmembrane protein, there are proteins which are present For example, again in RBC, hemoglobin is a protein. So protein plays very important role in biological function. These proteins are made up of amino acids, and there are 20 standard amino acids which will make up of all the proteins in the body. So when we say these proteins, they are linked. These amino acids are linked to one another with a bond. This is called as peptide bond, and this peptide bond we call it as a polypeptide chain. We call it as polypeptide chain. Okay. Now this single polypeptide chain, suppose there are 1, 2, 3, 4 codes present on the messenger RNA. There will be 1, 2, 3, 4 amino acids linked. There will be 4 amino acids linked to form a polypeptide chain. So, see, the genetic information has come from the DNA. DNA has transcribed into RNA, that is the messenger RNA, and then translated into a protein. How the amino acids has to be arranged in a protein, how the polypeptide chain has to be formed. This is the structure of a protein. So how this protein has to be formed? The whole coded sequence is present on the messenger RNA, and this is called as the central dogma of protein synthesis. That is, from DNA to messenger RNA, it is called as transcription. From RNA to protein, it is called as translation. So when the flow of information is carried, it is always in a single direction. It is always in a single direction. That is, it is unidirectional. It is unidirectional. So from the DNA, the message will be carried to messenger RNA. From the messenger RNA, the translation process, the coded language will be decoded, and the proteins will be synthesized. So every time a cell has to synthesize a particular protein, the genetic code, the DNA will have all the information. The DNA will suggest which protein has to be synthesized. The message will come from the DNA into the cytoplasm. The synthesis of protein is carried out in the cytoplasm. Why cytoplasm? Because the machinery for the synthesis of DNA for the protein, it is present in the cytoplasm. That is the ribosomes. Ribosomes are called as protein synthesis machinery. They are present in the cytoplasm. This is the ribosome. Messenger RNA. When the protein will come, synthesize, messenger RNA, protein synthesized, it will be released in the cell, and it will be carried out wherever it is required in the body. Wherever the biological function of the protein it will be carried out. So we have learnt about the central dogma of protein synthesis, which was given by Watson and Crick. Watson and Crick has suggested the central dogma of protein synthesis. But as I told you before, that there are certain viruses which have RNA as a genetic material. Now in this case, from only RNA, the protein cannot be synthesized. Why? Because this RNA does not have the required codes or the required information for the synthesis of a protein. It should always come from the DNA, and hence the RNA will be transcribed, and the process is called as reverse transcription. It is called as reverse transcription to DNA, and then the transcription and translation will carry out. So when a virus infects any host, then the virus has to make its own proteins. How it will make? I said these viruses, the genetic material of virus, which is RNA, it will be reverse transcribed into DNA, and then the whole process will again continue as DNA to messenger RNA and protein. So this concept was given by Temin. It is called as Teminism. Scientists named Temin and Baltimore, they have suggested this modification of central dogma where reverse transcription is also possible, where RNA makes the DNA and from DNA the proteins are synthesized. So I think you understood the central dogma of protein synthesis, which is very, very important when you learn molecular biology and biotechnology. So thank you, students. काय माहिती बंद नाही होत आहे. हे ब्लिंक करून करून बंद व्हायला पाहिजे ना. असं थोडी असतं. हो मग काय करू ते कोणच नव्हतं. पण हे बंद काय नाही होत आहे. बंद झालं नाही. काय माहिती बंद का नाही होत हेच नाही कळत आहे. इथे होते का बर्गर सर. हे असंच होतंय. ते ब्लिंक ब्लिंक होऊन बंद व्हायला पाहिजे तर होत नाहीये. पण हे ब्लिंक ब्लिंक होऊन बंद होतं म्हणाले ते होतच नाही. काय मला काही कळत नाहीये.